Hope everyone's got the energy for one more round. And this time again, modeling. And so now we're looking at an app for chronic viruses. And also I'll show you how to use it, not going through the graphical interface, but instead one step closer to the actual code. We looked at it briefly earlier, and now I want to actually go through that. First, real quick again, we load it, and then I'm going to actually go into the graphical interface real quick. And the reason why I'm doing it is such that we can look at the model real quick before we then basically leave it and look at it. So this is the chronic virus model. And if you look at the actual model, you see it has the same components again. We have uninfected cells, infected cells, free virus. We have an innate and an adaptive response. So in that sense, very similar. So what makes it a chronic model? So one thing is that for any kind of chronic infections, you need some kind of replenishment of resources. That's sort of a general idea. And in this case here, the uninfected cells, you have generation of new uninfected cells. For some pathogens, like as you learned for HIV, once the virus is actually in the DNA of the cell, actually you can sort of get replication of the virus by replication of infected cells that are latently infected. But in this case here, the main source will come here. So you see this is new here compared to earlier. We have now in the diagram here, the N, that's the rate at which new uninfected cells are produced, and then the DUU, which is just a natural way at which they can die in the absence of any kind of infection. So we have sort of a natural turnover, generation of new cells, and that's what would allow us to establish a chronic infection in the model. Also, if you look at the details here, the model for the innate and the adaptive response are now slightly different. So remember earlier, the adaptive response was assumed to be proportional to the innate response and then some rate and itself. So it was some RT times F. Now here I changed it now to the virus. So I'm saying the adaptive response is triggered proportional to how much pathogen there is. Either one is reasonable. Either one is probably not completely right. It's an approximation. If you have more pathogen or antigen load, that will lead to more probably activation of T cells. And also if you have more innate. So, why did I pick one in one model and why another one in the other? No particular reason in this examples. These are abstract teaching models. I just wanted to show you slight variations on the same theme. Now, if you build a real model for your system, you have to decide which is the most realistic or best approximation to model whatever you have in mind. Or, of course, again, if you're not quite sure, you can do it both ways and then compare the models and see what differences they give you. And based on that, decide which one might be more reasonable or realistic. So slight differences in the model. So then here we have the task again. And as always, feel free to any questions, just unmute yourself or type it in the chat and ask. Now here we have the instructions again, and we could do everything like we just did through the graphical interface, right? So read through this, we set things here, and then we do the run simulation, and we get our results. That will work perfectly fine here. However...
Okay, to let me know if I can be of service.
Okay. I don't know why Google is talking. That would work perfectly fine here. However, as you'll see down here, when we go to further ones, I'll ask you to do something for KT equals 10 to the minus 2, 10 to the minus 3, 10 to the minus 1, all the way, 11 different values. You can do that. You can adjust it, hit the button, record it, adjust it, hit the button, record it. But this kind of tedious thing is where computers are really shining. And then, instead of having you do it manually multiple times, you can just write a little computer code and let the computer do it automatically. So, and so usually, this graphical interface is nice for learning and exploring, but if you're doing a real project, you probably will want to do that writing code. And so that you get somewhat familiar with it, especially if you've never seen or done code before, that's what we're going to do now by writing a little bit of code to get things going here. Now, to do that, the thing is, if you have the graphical interface open, you see R here is blocked, so you can't actually do anything there. So what I'm going to do is I'm going to quit out of this one here. And so, it's now gone. And I'll interrupt it here. So now I'm back in the R console. Now the problem is I want to at the same time be able to read it. So there are two options. I could open another RStudio, and in one RStudio have it interactively running. So I see it on one side, and in the other one, I would then write my code. Another option, what I'm going to do now is if you go onto the website, you will have maybe seen that that there's an online version that I have on a website where you can actually interact with it online. In general, I recommend you install it because you have more flexibility. But now what we can do is we can go into the chronic virus model here and sort of basically read along and then do it over here. You can go to that same web page. So you could go to the main DSAIRM package website and click on the thing where it says, if you want to try it out, go there. That will get you to this web page here, or just type that into your browser. And then you could have it here at the same time, or you could have two RStudio sessions open. So if I want to know what underlying function I need, again, this is in further information. So here it's now called simulate chronic virus. Earlier it was called simulate acute virus. So I might, as a first step, go over here and quiz it and get the help file for it. And so here's the help file for this one here. Very similar inputs, and output will look very similar as well. So now we're going to do the first one here, but we'll do it with code. Now, you could write every command down there into the R console one at a time. That's usually not that great an idea, because then when you close R, it's all gone and it's hard to go back. So what one generally does, as soon as you start doing any kind of serious coding, you write it into a file. And then in the file, you just have all the commands. The good thing is you come back the next day, you open your file, you can just rerun everything and it's all there again. So that's, for any kind of serious code, that's what you're doing here. So also very, very highly recommended, comment your code. I always tell students at least 50% of your code should be comments, so you know what's going on there. It might seem overkill sometimes, but either your collaborator or your future selves will be very thankful if they look at your code and try to understand what happened there. So add a lot of comments, and in R, you add a comment with that hashtag, and then anything you write behind it, that's a comment. So I'm just writing something. And, feel free to code along on yours, write it to what I'm doing here, those steps. So to get this thing, you go to File, and then you say New File, a new R script, and this empty thing comes up. You see it's initially untitled, so at some point you want to save it to somewhere. I'm going to not just save it. Just somewhere, and so I have it when I want to come back. So you want to assign things to something, I just call it now res. So if I ran this would work. And I can take it, copy it down there. That's one way. The other option is I can just hit the run command here. That will just run this one line. Or if I have multiple lines marked, it would run them all. Or if I do source, it basically runs the whole thing. So those are different options you can do it. So now you see here in res, it showed up here, and it is, again has the structure that we've seen before. This data frame You might remember or not that earlier it was 501 observations of 6 variables. Now it's 2001. The reason is we run it for longer. Also, if you change the time steps, it would be different. So that's basically because every row here is 1 time step. That's why, depending on how you adjust your time, it might get longer or shorter. So that's with the default settings, but we might not want the default settings. It's always good to show what you're actually running it. So, oops, I don't want to type too much, so I'm just going to copy this bit from here. So these are the defaults. These are just the default settings for the different ones. And these directly correspond to these inputs in the graphical interface, right? We set those earlier graphically, we put numbers in the fields, and now you're just setting them right here. So now we want to adjust it so they correspond to what the app says we should be doing. So, basically now we want to do this. And instead of now implementing this instruction up here, putting it in there, we're now implementing it here. We're putting the numbers in. Now here you see there's only the abbreviation, not the meaning. So, if you can't remember what, let's say, DV was, you might want to go back here and find out, okay, that is the rate at which virus is cleared. And then you adjust it. So try that now. Just, read through this and then adjust the numbers in here accordingly, such that they correspond to what you had there. I guess there's one tricky bit that I'm just realizing now, that when you run it, you might not know how to actually pull it out. So come to think of it, maybe I should show you how to do that first. Otherwise, if you have some R experience, you would know how to do it, but otherwise maybe not. So let's just do the first one, and then you can do the next one, hopefully, then having the tools. I'm cheating a little bit. I'm just copying myself this over from the solution. So I don't have to just type it all in. But so basically, these are the settings. It doesn't really matter if you list it as one long spaghetti line or all underneath or mix like I do it here. It's as long as you comma separate it and close it with your parentheses, you'll be fine. So, and if you miss a comma or something and you try to run it, you'll get a problem error. Let's say I forgot this comma and then I'm trying to run it. One nice thing in RStudio is it kind of checks the code for you. And if it sees something that it doesn't think it looks right, it actually gives you an error message here already. You see the red X here. So that's nice, but I say I didn't see that and ran it, and you see I get some crazy error message. Often, unfortunately, our error messages are not the most informative, but after some time you get the hang and you know where a lot of the mistakes happen, and forgetting a comma or closing a parentheses or something that happens often. So we fix it now, and now it runs again. Now, conceptually, we know what we want, right? We want the final value. So the question asks, what the final value is. Steady state, that's basically at the end for the virus level. So we're interested in this virus variable here, and when we want the last entry. So, if you know R, then you know how to get in there. If not, that's maybe unclear to you, but basically you have this, what's called a list structure, this rest thing, and in that list structure is an element called TS for time series. And that TS has all these things in there. So for instance, this is the virus column. I could just print that on the screen. It now will print, 1,000, and then it said that's as much as I can print, so it doesn't print the rest. So really, I just want the end of it, not all of it. So in R, there's a command called tail, and so these are now the final ones. So this is a way, I can see now, yeah, these are the numbers I want. So going back to a question that came up earlier, how can you give what you have here to your collaborator? So when you run it here, you get the full time series of everything. So you can do whatever you want to do, not just what you can do through the graphical interface. So that's one nice flexibility you have here that otherwise you don't have. You can pull it all out. that also means, you can now make any plots you want. So, you can do the plot like we did earlier using the basic plot. Oh, okay, I renamed them. In the example, it's called result. Here it's res. So here you see it goes down, and then it sort of comes back up because the uninfected cells, that's what we're plotting here, are reproduced, they're replenished, and then it settles down in the steady state. If we wanted to plot the virus load, we could do that too. This is, we get a spike, and then it settles down to the steady state. So here we could do any kind of plot. This is using the base plotting commands that come with R. A lot of people these days like ggplot, which is what the package uses. If you wanted to create figures that look similar to what you see in there, you could actually use two helper functions. And there's a function called generate ggplot, and another one is called generate text, and each one of them basically create what's written underneath. So there's this weird list command. The reason is that the way these functions need it, they need a nested list. That's just so the underlying DSAIRM works. So in this case, you just take the result and stick it into another list, and then when you run it, you basically get the plot that you would otherwise get through the graphical interface. And down here, not very nicely looking, but on the screen, it writes you all these values that you see written underneath. So that gives you what you see on the screen. But again, the nice thing is that here now we can pull out anything we want. So instead of relying on this way of doing it, we can also do something like what I just did here, with the tail value here. Now, what we're being asked here is number of uninfected cells at the end of the simulation and number of infected cells at the end of the simulation. So, we could go here, res ts uninfected cells, and then again, tail. Now, tail by default pulls the last 6 out. And we don't need the last 6, we just need the last. So you can do 1 that just pulls out the last. So this gives us the very last one at the end. So you see this is the final value, and we could do that now for the infected cells as well. Now we can. You could assign that to variables. That is often good. And then you could print them if you wanted them printed on the screen. So that's how you write a little bit of code. Now, if you've never done any R coding, then even these commands are like, oh, what's going on there? But it's really not that complex. It's just some commands you need to learn, such as, one thing is how do data structures look like in R, like this list that we get back, and how do I dig into that list to get out what I want? And then commands like the tail command to get you the end, or the print command to print it on the screen. So that's sort of the downside, but the upside is that have much more control. You don't need to go through the graphical interface. You can now write this file and share that with someone else, and they could potentially just rerun it. In fact, to fully rerun it, they need to load the package, because this function here lives in the DSAIRM package. So if you didn't load that and they didn't have that installed, they couldn't rerun it. But if they have R, they install that package, you can give them that code, and they could rerun the whole thing and just reproduce it the way they want to. And they wouldn't have to go through the graphical interface. The problem with the graphical interface right now, and that's the problem with all graphical interfaces, if you've ever used any kind of stats program like Prism or so, when you do your clicking and so on, often it doesn't keep a trail of what exactly you did. And so it's not reproducible. And there's more and more push in science to make things as reproducible as possible. And if you do it by writing your code line after line, it automatically becomes reproducible, because it's clear what you did and someone else should be able to rerun it. So that's a big advantage. So here, I'll let you do now task 2. Why don't you try it? So the idea is now you change something here, double the rate of uninfected cells, then n to a different value, and then, run it again. And now instead of going through the graphical interface, what you do is you basically take this line of code. Maybe we should label them. Task 1. And then here's task 2. You take that line of code, you change these settings here, the ones that correspond to the instructions, and then you just run it again. And again, you can pull out what you need to. So try do these adjustments here, and then run it again. So let me do it here so you can see what we're trying to do. Double that one. So instead of 1, just 2 times 10 to the minus 4. So easy enough. We should do it here. I could keep calling it res. If I, for some reason, want to keep the old one around and later go back, I might call it something different. Basically, when you call it the same thing, and you're overriding the previous one. That is okay if you don't want to keep it. If all you want to save is uFinal and iFinal, and you don't care about the whole rest thing. Also, of course, you can always rerun it. But if you wanted to keep that, you could assign it a different name, and then you would have now res and res2. So we can run it here, and then we can also just sort of copy that code block here, move it down there. Now we have to replace here res with res2. So then we have to run it. Okay, maybe we'll look at the plot first. Looks slightly different. Let's also do the final virus here, so we can look at that one too. So these are the final values for each one. Now we've assigned it to these variables, but we haven't, printed them to the screen yet, so we can do that. What does it look like? I guess you can't push them all together. Okay, if we compare that to what we had above, I guess it would be good to call this 2. So we have them all separate. And do that same here. Just to make sure I'm running it again from the top. This one. Now I get these 3. I can print those, and then I get this one. Did something wrong? Yeah, I didn't pull out V here. Okay. Let's try that again. Okay, 3 different values. Do this one. I did it in a bit different order, but uFinal here is this uFinal, so that's the same. And iFinal and vFinal have changed. So let's see what we're supposed to do. So we're having more uninfected cells being produced. So, you could think about, well, if there's more uninfected cells that are constantly produced, then at steady state, there should be more of them. But this is one thing that's maybe not clear why you're getting what you're getting. So that's, a very simple model, but even for simple ones, sometimes intuition isn't good enough. And you want to then run it in a simulation to see what happens. And so basically what happens here, more uninfected cells just means more resources for the virus, which infects more cells, which actually then leads to an increase in the infected cells in the virus, but not an increase in the uninfected cells. So maybe something that, if you thought about it carefully, you might have, concluded it, but sometimes not. And then if you run a model and you see the result, then it often helps to think about it. Sometimes, once you've fully thought through it, you don't actually need the model anymore, but the model can often help you to think clearly through why is this happening? Why is that happening? Okay, so this was one example here. Now, I'm actually going to jump over a few because I want to get to an example where you can see where this additional way of doing it is useful. So in this one, we're just, turning on the immune response. But we'll hop to task 4 now, because now it kind of gets interesting here. We'll do the settings as are there, but then we run the model for all these different values for KT. Now, I could, of course, take that line of code and just make 11 copies, or however many it says, 11 different values. Make 11 copies of that code where everything is the same call. The only difference is that KT value at each time I'm calling it. That seems very tedious. And there's sort of a general rule in programming, which is called the DRY principle, which stands for don't repeat yourself. So the idea is if you do something more than like 2 times, you should find a way to write code such that you can reuse the same code multiple times. And one of the most basic structures that pretty much any programming language has is a loop. And the loop basically says just go over this piece of code multiple times, and every time either do exactly the same thing, but quite likely every time do something slightly different. And so that's what we can do here. We can actually write a loop around this statement and run the simulation. But each time we put a different, whatever it was, KF or KT in there, a different KT. And for each time we loop it, we record the result. And then in the end, we have the results we're interested in for everyone. Now, the amount of sort of simulated data we're getting is getting pretty big, because each time we get this whole time series of 6 variables and 2,000 each time. If for some reason we decided we needed all these time series all the time, we could save them all and take them along. But quite often we're not actually fully interested in the time series. We're interested in certain features like the peak or the final value or things like that. So then we just pull that out and record that. And if you know how to write loops, you can try to start, but since I assume not everyone is familiar with that, I'm not going to give you a head start, but just go ahead and do it. And again, I'm going to cheat and just copying from the solutions. And I'll share those solutions with you as well if you want to go back and take a look. So here's task 4. And so I'm copying first over, and then I'm going to walk through it. So here first, what I'm specifying all the different KT values that I want. So, I said I wanted from 10 to the minus 2 to 10 to the minus 12. All the way to this one. So these are the values I want to run for. So I'm just defining here a vector. Again, seq for sequence is another command in R. It just says make a sequence from 2 to 12 in steps of 1. And so on. Then I'm just having 10 to the minus 2, 3, 4, 5. And this is stored in this KTVec. So if I just run this command here and write KTVec here, you see it has these values in it. And then I'm setting up 2 vectors that right now I just fill with zeros. They will store the ones I'm interested in. I don't want to store the whole time series for every simulation. What I'm interested here is the final value of the infected cells in the virus. So I just set up 2 empty vectors. So basically just repeat 0, and the length of this vector should be the length of that KTVec. So you see it has all those zeros in there. There's a general rule, I could have also counted how long that KTVec is and set 11 here. But there's sort of a general rule. You want to hard code as little as possible when you write code, because if you decide, okay, no, I actually want to go from 2 to 22, then you would have to manually go in and change all these values for these vectors. So if anything you can set up, you specify once, and then other things are computed quantities based on that. That makes your code much more automated, and you can easily change it in one place and everything else will then change. And so then this bit here is sort of the major interesting part. This is this loop here. So I'm basically having a for loop that runs over 1 to length of this vector, so 1 to 11 in this case. And for each time it runs over it, it pulls one of these values out of that KT vec. So first it pulls out the first one, and the second, the third. All the other parameters stay the same. So I'm just running the simulation for different values of this one. And so every time I run the simulation, it's stored in this res. And then I'm doing what you already saw I did earlier. I pull out the bit I want, which is the infected and the virus at the end. So with the tail command, I can pull that out, and I store that here. So then I basically have 11 values stored in here and another 11 in the final vector stored in here. So I can go through that, and see, it takes a second because now I'm running the simulation not just once, but I just ran it 11 times. And now in the end, I can plot it. So now you're seeing I'm not actually plotting time anymore. I'm going one level more abstract, right? So I'm running an underlying simulation, which gives me back a time series. But then what I want to extract from the time series, I just want a specific value, such as, you know, the final or the maximum or things like that. And then I extract that, and then I plot that one as a function of that parameter of interest. So figures like this one that we just produced are some that you see very commonly in modeling papers. There's some parameter of interest, for instance, here the rate at which the T cells kill, or whatever your scientific question you're interested in. And then you're basically sort of scanning over that parameter, choose different values, and you look at how some outcome you're interested in changes, such as the final, the chronic level of the virus. You could, plot the virus now too. So see what that does. Looks very similar because the infected cells and the virus are kind of sort of tracking each other. So that therefore the pattern at the final value looks very much the same. So this is, with this way of doing it now, very easy. You can easily produce those figures. A figure like the one we just produced through the graphical interface, you can't produce. There are two apps that let you do it. So those two apps that are called Exploration, they have this kind of thing built in where you can do it, but it's only for the basic virus model and the basic bacteria model. So for the chronic virus model, going through the graphical interface, we couldn't easily produce the figure we just produced. But doing it in this way, where we just call this underlying function multiple times, we can produce these kind of figures. So now, as you see, you have a good bit more flexibility by writing a little bit of code and calling the underlying functions. And of course, no one would prevent you from doing a double loop. Let's say you might want to loop over this parameter and you also want to loop over the P parameter. Then you could have one loop and you wrap it around yet another loop. It will start taking a little bit because, if you have one going over 10 values and the other over 10 values, then you have a 10 times 10. So you're running 100 simulations. But these simulations don't take too long. So even, shouldn't take more than a few seconds. And then if you loop over 2, you could, for instance, do a 2D heat map plot where whatever the quantity of interest, you give it a color. And then your X and Y axis could be some kind of, other two parameters of interest. Now, of course, you can go further. You can loop over 3 or 4, but at some point it gets unwieldy and you can't graphically represent it anymore. So generally what you would then do is you might then instead take slices. Let's say your main parameter of interest is this KT, but you're also secondarily interested how the impact of KT on the outcome is affected by changes in P. So then you might run your simulation for 3 values of P and put all these 3 lines into your figure down there. So you have one line for P equals 100 and another line for P equals 10 and one line for maybe P equals 1. Again, very common plots. So if you look in the literature, any kind of modeling papers, you see these kind of plots very commonly. Also, again, just I'm sure you realize it, but there's no data here, right? This is all just simulations. So we're building our model, for any real setting, we need to pick the parameters based on what we know about the system. But then once you've built the model, you can explore it and run it without the need to fitting it to data. So again, this is very much in contrast to any kind of statistical model you're familiar with, where you have to have data to apply the model. They just go hand in hand. Okay, let me see if there's any other interesting thing going on. Here's another example. Of course, then you can go, say, okay, I want this scanned much more finely. So instead of only doing 10 values, I'm actually now doing 100 values. So you can do that too. Now, of course, the downside is the whole thing will run longer. And you see it's still running, but you see I didn't have to write any more code if I do it 10 times or 100 times. If you had to copy and paste it, or in the graphical interface trying 100 different values, that would be very tedious. You didn't want to do that. Here, the only thing I have to do is just wait a bit longer, which I just did. And so now you see I have a much more nicely smooth curve because I'm trying many more values that I can do here. Okay, I don't think in this one I'm doing any kind of oh, actually, there's an example here. So I might not copy over the code for the sake of time, but I'll share it with you. So if you go to task 6, you see I'm asking you what I just described. I tell you scan over that KF vector. Now we switch to the KF instead of the KT. And then I said, well, we also want to know how the outcome, in this case, the outcome we're interested in here is the peak of the virus load, how that depends on, KF for different values of that RT vector. So, here, that's what we're doing here. We put them both in there. And you see the curves are crude, but you could, of course, run them for more points, and then you get a nice smoother one. And so you get something like this. So now here you can see an example where, the impact of that KF parameter on the final virus load is actually very different based on the value of another parameter. So, these kind of behaviors, even for a relatively simple model like this one, you can get interesting things. And then, you probably want to dig deeper and see what does it mean, and, which one is biologically more realistic, this behavior or that behavior. But so you can do all of this here with a little bit of code. And so that's what I call generally the level 2 approach, where you call these underlying functions that are in the package and you can explore them. And trade-off, a little bit of coding. The advantage is much more flexibility and much more closer to actually now, starting to actually model. The graphical interface is good for learning, but it's not actually, doing your own modeling. Doing your own modeling always pretty much means some level of code writing. And so this is starting to move you into that direction. Depending on how familiar with it, this could be a very, familiar and you've seen it all, or it could be very new and it would take you some time to learn the coding. But, there's lots of good resources out there that teach you languages like R. Of course, we can't cover coding in 2 days, really. But, if you're interested in that, there's lots of good resources out there. And at that level of coding, it wouldn't take you too long to pick it up. If I teach it over a semester, usually by the end of the semester, students are pretty good at writing code at that level. Okay, and I think I'll stop the walkthrough at this level. We have a bit more time for any questions on this or generally on the modeling.